Hello and welcome to Easy Learning with Dr. Salman Khan. In this video, we will learn how to study a parasite. This is the format under which we will study a parasite. Introduction, Epidemiology, Habitat, Morphology, Life Cycle, Modes of Infection, Pathogenesis, Clinical Manifestations, Laboratory Diagnosis, lastly Prophylaxis and Treatment. So first, let's begin with Introduction. Introduction comprises of two parts, Name and History. Under Name, we study the genus and the species to which the parasite belongs. So usually a parasite has two parts in its name. The first part comprises the genus of the parasite and the second is the species to which it belongs. Next, History. Under History, we study the name of the discoverer of the parasite and the place where it was discovered. Thirdly, Epidemiology. Under Epidemiology, we study the geographical distribution of the parasite, that is, throughout the world, which are the areas where it is commonly found. Next is Habitat. Under Habitat, we study which are the areas of the host where the parasite infects. That is, which are the organs where the parasite causes infection. Certain organs will be the primary focus of infection, whereas certain organs will be the secondary focus of infection. Next is Morphology. Morphology refers to the structure of the parasite, the physical appearance of the parasite and the structure it is made up of. Next is the Life Cycle. The life cycle of a parasite can be of two types. One is direct life cycle and indirect life cycle. Direct life cycle refers to when a parasite requires only a single host to complete its development. Example, entamoeba requires only a human host to complete its life cycle. Whereas indirect life cycle is when a parasite requires two or more species of hosts to complete its development. Example, malarial parasite requires both human host and mosquito host to complete its life cycle. Next, the modes of infection. Modes of infection is divided into three parts. First, sources. Second, portal or the route of entry. Third is the vehicle of transmission. First, the sources of infection. These are the following sources of infection: contaminated soil and water, food, vectors which are divided into biological and mechanical, carrier, and lastly self-auto-infection. The first, contaminated soil and water. Soil polluted with embryonated eggs containing roundworm, whipworm, etc. may be ingested, or infective larvae in soil may penetrate exposed skin. Example, hookworm. Infective forms of parasites present in water may be ingested. Examples, cyst of amoeba and giardia. Water containing the intermediate host may be swallowed. Example like cyclops, which contain Guinea worm larva. Infected larvae in water may enter by penetrating the exposed skin. Example, cercaria of schistosomes. Free-living parasites in water may directly enter through vulnerable sites. Example, Naegleria may enter through nasopharynx. Food. Ingestion of contaminated food or vegetables containing infective stage of the parasite. Example, amoebic cysts, echinococcus eggs. Or ingestion of raw or undercooked meat harboring infective larvae. Example, pork containing Cysticercus cellulose, the larval stage of the parasite Taenia solium. Vectors. A vector is an agent, usually an arthropod, that transmits an infection from man to man or from other animals to man. Example, female Anopheles is the vector of malarial parasite. Or these vectors can be biological vectors. Biological vector. The term biological vector refers to a vector which not only assists in the transfer of parasites, but the parasites undergo development or multiplication in their body as well. They are also called as true vectors. Example of true vectors are mosquito in cases of malaria and filariasis, sandflies in cases of kala azar, tsetse fly in case of sleeping sickness. Next, mechanical vectors. The term mechanical vector refers to a vector which assists in the transfer of parasitic form between hosts but is not essential in the life cycle of the parasite. Example of mechanical vectors: housefly in case of amoebiasis. Then animals, domestic animals, that is cow, example Tinea saginata infection, pig, example in Tinea solium infection, dog in case of Echinococcus granulosus. Next, carrier. Carrier is a person who is infected with parasite without any clinical or subclinical disease. He can transmit parasite to others. For example, all anthropoonotic infections, vertical transmission of congenital infections. And lastly, self or auto-infection. Example is finger-to-mouth transmission in case of pinworm infection. Then internal reinfection in case of infection by strongyloids parasite. Next, portal or the route of entry. These have already been discussed in detail under the third section, transmission, which has been covered in the previous video. Next, vehicle of transmission. These can be through insects. Example, mosquito, sandfly, ticks, housefly. And animals, example cow, dog, cat. Humans, a human can be a carrier with clinical or subclinical disease. And lastly, fomite, example bedcloth, towel, etc. So these are the modes of infection. Next, pathogenesis. Pathogenesis is divided into inapparent and clinical disease or infection. Clinical disease comprises of pathogenic lesions, which are again subdivided into macroscopic and microscopic. Macroscopic refers to the lesions which can be seen with the naked eye, whereas microscopic lesions refer to those lesions which require the help of a microscope to identify them. Clinical infection is divided into acute infection, subacute infection, chronic infection, latent infection, and recurrent infection. Next, clinical manifestations. These are divided into objective symptoms, also called as signs, and subjective symptoms. Next, laboratory diagnosis. The diagnosis of a parasite can be done through the following ways: by microscopic examination, through culture, serological tests, skin test, animal inoculation, molecular method, xenodiagnosis, imaging, and through hematological examination. First, microscopic examination. It includes stool examination, blood examination, urine examination, sputum examination, cerebrospinal fluid or CSF examination, tissue and aspirates, and genital specimens. First, stool examination. Examination of stool is very important for the detection of intestinal infections like Giardia, Entamoeba, Ascaris, and Cyclostoma, etc. Cysts and trophozoites of Entamoeba histolytica, Giardia lamblia can be demonstrated in feces. Eggs of roundworms and tapeworms are also found in stools. Second, blood examination. Examination of blood is of vital importance for demonstrating parasites that circulate in blood vessels. Malarial parasite is confirmed by the demonstration of its morphological stages in the blood. Next, urine examination. The characteristic lateral spined eggs of Schistosoma hematobium and trophozoites of Trichomonas vaginalis can be detected in urine. Microfilaria of Wuchereria bancrofti are often demonstrated in the chylous urine. Sputum examination. The eggs of Paragonimus westermani are commonly demonstrated in the sputum specimen. Occasionally, larval stages of strongyloids, stercoralis, and Ascaris lumbricoids may also be found in the sputum. Cerebrospinal fluid examination. Some protozoa like Trypanosoma brucei, Naegleria, Acanthamoeba can be demonstrated in the CSF. Next, tissue and aspirates examination. The larvae of Trichinella and eggs of Schistosoma can be demonstrated in the muscle biopsy specimens. In Kala Azar, Leishman-Donovan or LD bodies can be demonstrated in the spleen and bone marrow aspirate. Trophozoites of Giardia can be demonstrated in intestinal aspirates. Trophozoites of Entamoeba histolytica can be detected in liver pus in cases of amoebic liver abscess. Then genital specimen examination. The trophozoites of Trichomonas vaginalis are found in the vaginal and urethral discharge. Eggs of Enterobius vermicularis are found in anal swabs. Next, culture. Some parasites like Leishmania, Entamoeba, and Trypanosoma can be cultured in the laboratory in various media. Then, serological tests. These are helpful for the detection and surveillance of many protozoal and helminthic infections. These tests are basically of two types. First, tests for antigen detection. Second, tests for antibody detection. First, antigen detection. Malarial antigen like Plasmodium falciparum lactate dehydrogenase, which is also called as PLDH, and histidine-rich protein 2, also called as HRP2, are detected by rapid immunochromatographic test. Filarial antigens are detected in current infection by enzyme-linked immunosorbent assay, commonly called as ELISA. Next, antibody detection. The following antibody detection procedures are useful in detecting various parasitic infections like amoebiasis, echinococcosis, and leishmaniasis. That is, the tests like complement fixation test, that is CFT, indirect hemagglutination, IHA, indirect immunofluorescent antibody, IFA test, rapid immunochromatographic test, ICT, and enzyme-linked immunosorbent assay test, commonly called by the term ELISA. Next, skin tests. Skin tests are performed by injecting parasitic antigen intradermally and observing the reaction. In immediate hypersensitivity reaction, wheal and flare response is seen within 30 minutes of infection, whereas erythema and induration seen after 48 hours of injection is called as delayed hypersensitivity reaction. Some important skin tests which are done are Cassoni's test in hydatid disease, Montenegro test or Leishmanin test done in Kala-Azar, Fairless test done in schistosomiasis, and Bachmann intradermal test done in trichинellosis. Next, animal inoculation. It is useful for the detection of Toxoplasma, Trypanosoma, and Babesia from the blood and other specimens. Next, the molecular methods. Molecular methods most frequently used to diagnose human parasitic infection are deoxyribonucleic acid, that is DNA probes, polymerase chain reaction or PCR, and microarray technique. These tests are very sensitive and specific. Next, xenodiagnosis. Some parasitic infection like Chagas disease caused by Trypanosoma cruzi can be diagnosed by feeding the larvae of redovoid bugs with the patient's blood and then detection of amastigotes of Trypanosoma cruzi in their feces. Next, imaging. Imaging procedures like X-ray, ultrasonography, computed tomography, also called by the term CT scan, and magnetic resonance imaging, that is MRI, are now being extensively used for diagnosing various parasitic infections like neurocysticercosis and hydatid cyst disease. Then hematology. Anemia is frequently seen in hookworm infection and malaria. Eosinophilia is frequently present in helminthic infections. Hypergammaglobulinemia occurs in visceral leishmaniasis, and leukocytosis is seen in amoebic liver abscess. These are the various laboratory diagnoses done in order to detect the parasites. And lastly, prophylaxis and treatment. Prophylactic measures are to be taken based upon the type of infective source, route of entry of the parasite, and the method of its transmission to avoid getting infected. If in case infection happens, the treatment now depends upon the type of signs and symptoms produced by that parasite. So this is all about study of a. If you wish to see more of such videos, do comment below. And don't forget to like, share and subscribe to my channel. Also, press the bell icon for early notification of my new videos.